So in this module, we're going to have our first, I guess, discussion of metabolism. And this is, I guess, one of three, the three-part discussion of metabolism. In this first, in Module 10, we're going to be, let's just go to our learning targets here. We're going to be talking about, again, just some terminology and definition, again, to kind of refresh our memory about what energy is, giving us a biological definition for metabolism, talk about anabolic catabolic reaction and metabolic pathways. We'll get into talking about enzyme and their functioning. We'll get into talking about oxidation reduction reactions and their connections to within metabolic pathways. And then we'll look at the structure and production of ATP. So let's start with, again, some definitions here. Should be just probably a review for you. You're well aware of different types of energy. Remember, energy is the ability to bring about change or do work. So that's kind of our physics definition. So there's a variety of things that fall under that energy definition. And again, light energy, heat energy, chemical energy, electrical energy, nuclear energy, mechanical energy, potential kinetic energy, and so on. So, and remember that living organisms use chemical energy, and that chemical energy that living organisms use is found in chemical bonds. It's bond energy. So keep that in mind. And when we define metabolism, usually, we say metabolism, you think of like the reactions that we, breaking down food to make energy for the cell, right? Making ATP. But remember that when we talk about metabolism, we're talking about all the biochemical reactions, all the biochemical pathways that go on to maintain life functions within the living organism. When we look at those metabolic reactions within living organisms, we can separate them into two groups. We have catabolic reactions, and those are reactions where we're taking larger compounds and breaking them down or removing things from larger compounds. And when we do that, in most cases, we're releasing energy. Catabolic reactions are energy being released. They can be used to provide building blocks, provide those basic units that are needed to make more complex molecules, like our macromolecules. Then the second type of reaction then are anabolic reactions. And these are reactions where you take and build compounds. So you take those basic units, those building blocks, and make more complex molecules. So for example, if we were looking at making our macromolecules, you take amino acids and make proteins. That would be an anabolic reaction where we're taking those basic units and making amino acids to make proteins. And again, notice that these are using energy. Anabolic reactions require energy. And so these reactions, the catabolic and anabolic reactions, are tied together. They're coupled. They're connected together. And of course, the energy that's needed, or that's being used, for those anabolic reactions is ATP. And then energy being released by catabolic reactions can be used to make ATP. So if we look at our little cartoon here, we see that we take these large complex molecules, and if we were ingesting those in our food, and we digest those down, then we're doing catabolic reaction. We're doing catabolism. We're taking those starches and breaking them into simple sugars and proteins and amino acids and so on. Then when those smaller units are produced, then some of those smaller units are going to go into down in our cartoon here and go into cellular respiration, be further catabolized to make ATP. And of course, sugars, simple sugars, complex sugars can be digested down, catabolized. Simple sugars aren't going to be catabolized any further until they go into cellular respiration. But let's say we have starch, we're going to first break that down into sugars, and then the sugars can go into cellular respiration, right? Make ATP, right? And then notice if we go up here from our basic units, then we're doing anabolic reactions where we're taking those basic units and along with ATP energy and making the more complex macromolecules in this case. So taking those glucose molecules and making starch, or taking amino acids and making protein, and so on. Okay. So again, these coupling reactions, the energy from catabolic reactions is used in the form of ATP to make to do the anabolic reactions where we make those more complex molecules. So in living organisms, both catabolic and anabolic reactions, in some cases, they may need a lot of energy or they may release a lot of energy. So, and you may not be able to supply that energy all at once, or you may not want a lot of energy released from that reaction. It could be dangerous, for the cell. And so what we find is that in living organisms, these metabolic pathways occur within a series of linked reactions where you start with your beginning product of your beginning reactants, your raw materials, and then in a step-by-step manner, you finally get your final product. And so the cartoon here is trying to illustrate that, where we have our first reaction, where we take the raw materials, they're changed or catabolized to produce the products, and then the products serve as reactants for reaction 2, and then the products of reaction 2 serve as reactants for reaction 3, and then reaction 3 we finally get our final product. The numbers in the arrows then are going to represent catalysts that help catalyze those reactions. And enzymes are those catalysts. So enzymes are proteins that have a specific three-dimensional shape that gives them biological activity, and then they are used in catalyzing the reactions in metabolic pathways. And we have an example. Here. Alcohol, and if the alcohol in the presence of the enzyme alcohol dehydrogenase will bind to that, and alcohol dehydrogenase will convert alcohol into acetaldehyde. And then if the acetaldehyde dehydrogenase enzyme is around, it'll convert acetaldehyde into acetic acid or vinegar. So when they were making wine long ago, there would be some of the wine would go bad. It would turn into vinegar, and they would get a contaminating bacteria that was doing this step where it was converting the alcohol in the wine to acetic acid using these two enzymes to do that. So eventually they were figured out what was going on there, and Louis Pasteur and some pasteurization helped kind of take care of some of that problems they're having with their wine going bad. So if we talk about enzymes a little more in depth here, we're going to go quite in depth, I guess. You see that their proteins have three-dimensional shape. They will average size approximately can contain approximately 300 amino acids, 10 to 1,000 Daltons. 100 Daltons equals 1 amino acid, 110 Daltons. Again, notice that they are involved in the chemical reaction. They're catalyzing the reaction, but they're not used up in that. They're able to lower the amount of activation energy or startup energy that's needed for the reaction to occur. They'll allow for access to dehydration synthesis or hydrolysis reactions that will catalyze these reactions in the metabolic pathway. If you'll find that if you see a word that has ASE, so that is probably an enzyme. That word is probably belonging to an enzyme or a name of an enzyme, but not always the case. And keep in mind that Enzymes are very reaction specific. So in that diagram that we looked at, the like for example, the alcohol dehydrogenase enzyme, that enzyme will only convert alcohol into acetaldehyde, and that's the only reaction it'll do. Um, and notice what talks about transferring carbon dioxide into the blood. So carbon dioxide will naturally diffuse in the blood based on partial pressures. But if the carbonic anhydrase enzyme's there, notice how much it speeds it up. It increases that reaction over 107 times faster than without the enzyme. Pepsin, so now that is an enzyme. It's a group of enzymes called proteases. Now notice that pepsin does not end in ASE. So there's an example of an enzyme that's not end in ASE, but it digests proteins, right? And it's produced in your stomach and helps digest proteins. There are well over 4,000, maybe 5,000 known or characterized enzymes, and they speculate that there may be up to 75,000 just in humans. So we haven't identified by far all the enzymes at all. Probably not even close. Again, some enzyme categories. So if it's transferase, it's an enzyme that helps transfer functional groups from one organic molecule to another. Amylase breaks down starch. Proteases we talked about, mentioned already, breaks down proteins. Ligase will join molecules together. So again, some different groups of enzymes. Group enzyme categories, excuse me. And then there'll be different enzymes in those categories. So if you look at enzyme structure, again, remember they have this three-dimensional shape that gives them biological activity. And there's a site on the enzyme where the reactants bind. The reactants are called substrates. And the site on the enzyme is going to have that the substrate binds, that the reactants bind to. It's going to have a three-dimensional shape that'll fit or allow the enzymes to fit in and bind to the enzyme. And they'll form that enzyme substrate complex. They found that a lot of enzymes, once the substrate binds, it actually kind of forms a little tighter fit. It's called an induced fit. So if the right substrate binds there, it even makes it an even tighter bond between the enzyme and the substrate. And then it'll catalyze the reaction. In this case, the one substrate is broken down into two products. And then the enzyme is released and can go right on catalyzing the reaction as long as there's more substrate there. So again, it's not a reactant. It's not used up during the chemical reaction. And this graph is trying just to illustrate the difference between a reaction that's catalyzed by an enzyme versus the same reaction that's not being catalyzed by the enzyme. Again, both reactions need activation energy. The red line is showing that the enzyme catalyzed reaction just needs a lot less activation energy. And then again, it's showing you that the products, the energy level of the products is still the same, no matter if it's catalyzed by the enzyme or not catalyzed by the enzyme. And again, remember that the bond energy, the chemical energy, is bond energy. So the bonds of a compound contain the chemical energy. Okay, so we're going to watch a video, and if I'm hoping you'll be able to hear that through as we go. If not, again, this presentation is posted. So if you're not able to hear the video play as I record it here, you'll need to please get on the presentation on Canvas and watch this. It's going to talk about some factors that can affect enzyme and enzyme catalyzed reactions.
When enzyme and substrate molecules are mixed, they randomly collide, forming enzyme-substrate complexes. As temperature increases, so does the kinetic energy of the molecules, and therefore also the rate at which these collisions occur. There comes a point at which the maximum rate of reaction is achieved, and the temperature at which this occurs is known as the optimum temperature. If the temperature continues to increase beyond the optimum, the enzymes begin to lose their three-dimensional structure and become denatured, therefore reducing the rate of reaction. Now you need to be able to recognize the graph of the effect of temperature on enzymes. It looks like this, with the peak being the optimum temperature. To explain up to the peak, as the temperature increases, so does the kinetic energy of the enzymes and the substrates, resulting in an increased frequency of collision between the two, thus increasing the rate of reaction. But beyond the optimum temperature, this continuing increase in kinetic energy results in a loss of 3D structure from the enzyme and therefore a change in its active site. If the substrate can't fit into it, the enzyme is referred to as being denatured, and this reduces the rate of reaction. Now for a look at pH. Increasing pH values are as a result of adding hydroxide ions, or OH-. Decreasing pH is as a result of adding hydrogen ions, or H+. Either of these can potentially alter the charges on the amino acids that make up the enzyme, and this can lead to the enzyme's active site changing shape because the bonding changes, for example, and that could denature it. Or it could mean that the substrate can no longer bind to the enzyme's active site, and so either way the rate of reaction is reduced. The graph for the effect of pH on enzyme activity is very similar to that of temperature, although you might notice a more symmetrical decrease in rate of reaction either side of the optimum pH. On to having a look at substrate concentration. When we change substrate concentration, we are assuming that enzyme concentration is remaining constant. Initially, increasing substrate concentration from 0 will increase the rate of reaction as the substrates randomly collide with the enzymes and the enzymes catalyze the chemical reaction. However, there comes a point where all of the enzymes' active sites are filled and the enzymes are already working as quickly as they can to catalyze the reaction. This is where the maximum rate of reaction is reached, and any further addition of substrate beyond this will not increase the rate of reaction anymore.
Okay, so this just touch base on a couple of things. So now remember with temperature, again, just the temperature increases a little bit or temperature decreases a little bit, that's just going to affect the rate of reaction because of molecular motion, right? If the temperature warms up, then the molecules, the molecules are moving faster, and so the reaction is just going to go faster. The enzyme substrate complexes are going faster, and so on. And then again, the opposite. If it gets the temperature lows, gets the molecules are going slower, and now, of course, there is a point if it's going too slow, gets too cold, then that can affect the organism, especially if you're not making ATP or things you need, right? Now, again, also, if the temperature gets too high, as you mentioned, then you're going to affect the enzyme. It's going to lose its three-dimensional shape. It's going to denature, and then that turns off, stops. The chemical reaction stops. The cell's dying. Things are, harming the organism, right? Now with pH, it's strictly going to affect the enzyme. So again, as long as you're within that pH range, everything's going fine. But if the pH gets too acidic, pH gets too basic, then the enzymes lose their three-dimensional shape and reactions stop, damages the cells or damages the organism, or they die, right? And then as she talked about with enzyme, the amount of enzyme and the amount of substrate. Again, if you keep the substrate the same, add more enzyme, again, it's going to increase the rate of reaction up to a point. It'll eventually reach that point to where the enzyme and substrate are making those connections as fast as possible. It's not going to go any faster. The same way if you increase the substrate there. Again, these graphs that she showed, similar to what she showed the video. Now notice again with the temperature, you have this gradual increase. As temperature increases gradually, again, the reaction rate is going to go faster. until it gets too hot. It gets over higher than its optimum temperature, and then it drops really straight because enzymes are denaturing. And again, with the curve for pH, the slope's pretty much the same because either too acidic or too basic, the enzyme denatures, the reaction stops. And again, with increasing substrate concentration, there'll be a point to where enzyme substrate complexes are forming as fast as they can, and so the reaction is going to peak and not go any faster. Again, just remembering that this is the structures of the protein, again, is directly connected to functionality. And so when we talk about enzymes, we're talking about, tertiary structure. We have these three-dimensional shapes that give the enzyme biological activity, and you have these active sites where substrates can bind and those types of things. And remember the quaternary structure that we were talking about when you put multiple tertiary structures together to form a functional protein. And that example there is showing hemoglobin, where you have the 4 heme molecules that are in a tertiary structure complex together to form that quaternary hemoglobin molecule. So in the study of enzymes, they found that there's certain things that some enzymes need for them to become functional. So some enzymes need ions, potassium ions or sodium ions. And so enzymes that need these ions, the ions are called cofactors. They're essential for allowing enzymes to function properly. If it's a non-protein organic molecule that is needed, then they're called coenzymes. And a lot of vitamins like B vitamins are coenzymes. And coenzymes here, trying to illustrate that coenzymes are function in forming the proper enzyme substrate binding site. So you have your enzyme without the coenzyme attached to it, and that's called the apoenzyme. So it's not active, it's an inactive form. And then it becomes active when that coenzyme binds and then forms the proper shape of the substrate binding site. And so now you have your holoenzyme or your whole enzyme, and it's active in that form. Some biochemical pathways need to be turned on all the time or function all the time, so that we say that they're constitutively expressed, and so that whatever it's making, that product's always being needed by the cell or by the living organism. And then there'll be other times when the metabolic pathway is turned off, so you have something that will induce, that turns it on. It's called an inducer that activates it. And then there's times when you have made enough of your product that you need to shut down that pathway. And so then you'll have repressors or things that inactivate or turn off the metabolic pathway. So a common way of turning off metabolic pathways is actually by blocking or turning off the enzyme, so to speak. So one way of turning off an enzyme or turning off a metabolic pathway is when the end product, is able to come back and loosely bind to the active site on the first enzyme in the pathway. And this is called competitive inhibition when it does that. It's competing for the substrate binding site on the first enzyme. And so as more and more of the product is accumulated, then it's turning off. It's turning off the more and more of the enzyme by outcompeting the outcompeting the substrate for the enzyme binding. A second way, which was actually shown on that cartoon there too, is non-competitive inhibition. So in this situation, again, it's the end product that is accumulating. Now this time the end product will bind not on the active site, but to another site that's called an allosteric binding site. And when it binds to that allosteric binding site, see it here in yellow, the inhibitor bound, and then it changes the three-dimensional shape of the active site, and so now the substrate cannot bind. And so again, as that end product accumulates, it binds up more and more, binds to more and more of the enzyme, thereby shutting down that pathway. And that's how cyanide works. It binds to an enzyme in electron transport chain, if I remember correctly, shuts that off and not make ATP, and it affects you very quickly. And then remember penicillin, when it prevents the cross-linking of newly formed peptidoglycan wall, it's doing that by binding to the enzyme that's involved in making those allowing those tetrapeptides, peptides, short peptides to cross-link. It doesn't allow that. And this type of regulation that we were just talking about, where we're turning off the metabolic pathway by using either substrate level or, using competitive inhibition or allosteric binding of the product, is a type of feedback inhibition. Again, the cartoon here on the right is showing we have our enzyme sets of catalyzed reactions. So we have enzyme 1, enzyme 2, enzyme 3, and then our product starts to accumulate after reaction 3. And as that product builds up, it starts to bind to an allosteric binding site, in this case, on enzyme 1. And when it does that, it changes the active site of enzyme 1 so that first substrate can no longer bind. And again, as this end product from reaction 3 continues to build, it starts turning off more and more of the enzyme 1 in that reaction and eventually stopping the pathway. Okay, so I have an e-ticket for you to do. I have it set up to where it's a student-paced lesson. So if you get onto your app or go to join.nearpod.com on the web and type in the Nearpod code there, the SXDNG, it will open up that Nearpod. There are 2 questions for you to answer. So please answer those before our next lecture. And that is the end of, we're not done with this presentation yet, but we're going to stop here for now.